Coconut is one of the important palm species in tropical and subtropical regions of the world and plays an important role in the socio-economic life of smallholder farmers in these regions. Despite its great economic importance and multitude of uses, genetic erosion of coconut continues mainly because of pest and disease attack and natural disasters. More than 80% of living coconut cultivars are only conserved in one gene bank, so there is a strong need to implement efficient germplasm conservation strategies before further loss occurs. Conventional storage as seed is not applicable for coconut because of the large size and recalcitrant nature of the seeds. Growing of plant in field gene bank and botanical garden need a very large area for the maintenance, and the process is highly costly. In addition, the conservation sites are always under the threat of adverse weather conditions and pest and disease attacks. Therefore, cryopreservation is the only promising method for long-term conservation of coconut germplasm. The aim of this study was to develop an efficient protocol for the cryopreservation of coconut zygotic embryo using rapid physical dehydration. Based on the previous protocol developed in our lab, the highest germination percentage of embryos was achieved in moisture content of 20% after 8 hours of drying. So the goal of first experiment was to improve the hydration protocol to dry embryo faster in order to minimize damage to embryo. For this purpose, embryos were pre-cultured in different sucrose concentrations for different days and then physically dehydrated for different periods of time using new developed drying apparatus. Based on the result, pre-culture of embryo in 0.4 and 0.5 molar sucrose for 5 days could decrease the moisture content of embryo to 20% only after 3 to 4 hours of drying with apparatus. In the second experiment, the effect of best sucrose pretreatment and dehydration times were evaluated on survival rate of embryo after cryopreservation. The survival of embryo after cryopreservation was only 20% in control treatment, while pre-culture of embryo in 0.5 molar sucrose for 5 days increased the survival rate of embryo to 60%.